In the last lecture, we talked about metabolism and the breaking down and digestion of food, specifically carbohydrates. So we broke down carbohydrates into smaller disaccharides and eventually monosaccharides, and eventually everything converged to glucose. So now once we have glucose, then what do we need to do? Well, we want to take glucose to energy. So glucose has to be further broken down in order to release the energy. And how this happens is through glycolysis. So the stage 2 of digestion is actually the cellular metabolism. It is the breaking down of glucose in the presence of oxygen to give carbon dioxide, water, and energy. So the first step is glycolysis. We'll go through that. And then there's pyruvate dehydrogenase. Then takes it into the Krebs cycle or the TCA cycle, and finally electron transport. So there are a number of different metabolic pathways that break down carbohydrates as proteins and lipids, and we'll talk about them individually. But some of these steps are what's called irreversible. We mentioned that they have a committed step and they cannot go backwards. So those will have different catabolic and anabolic paths. And you can see here, so if you have reactant 1 goes to A and then goes to reactant 2, when you want to go back, you can't just go 2 to A to 1. You have to have some alternate path. And the reason for this is because you don't want competing pathways taking place in the organism at the same time in the same place. So there are a number of metabolic enzymes, and they're the usual ones. They're oxidation reduction reactions, isomerization reactions, elimination reactions, more isomerization, and reactions that break bonds, such as lyases, hydrolases, and ligases. And we're going to talk about each of the enzymes as they come up. So right now we said we're going to talk about glycolysis. So glycolysis is a metabolic pathway that uses glucose. It degrades 6 carbon glucose molecules into 3 carbon pyruvate molecules in 10 steps. This takes place in the cytoplasm of the cell. It generates ATP, and again it is 10 steps. So we see the steps on the side. And I like this representation because this is about the level that you're going to have to know it at. I don't want to expect you to draw any of the structures, but know by name. Glucose goes to glucose 6-phosphate and what enzyme is going to be involved in that. So this is the level that we need to know. So what is the purpose of glycolysis? It is to extract energy from glucose molecules and other 6-carbon sugars and convert that into energy that we can use for cellular work. So that is in the form of ATP or NADH. It can also be used as raw materials for biosynthesis of other molecules, and we'll see where some of them go, including amino acids, lipids, and nucleic acids. So this is an overview. We said there were 10 steps, and these 10 steps can be divided into 2 phases. There's the energy investment phase, where 1 glucose is broken down into 2 glyceraldehyde 3-phosphates, and then those 2 go into the second phase, which is the energy payoff phase. And each molecule of the glyceraldehyde 3-phosphate is then oxidized into a pyruvate molecule. So before we get too much into it, I just want to talk about the location of where these things occur. Glycolysis occurs in the cytoplasm. So we did all the digestion, which were in your digestive organs. It is absorbed into the bloodstream, taken out to the cells, and in the cytoplasm of the cells is where the glycolysis of glucose takes place. Once it's converted to pyruvate, pyruvate then is transported into the mitochondrion where the TCA or Krebs cycle takes place. And we'll talk about that as we get to that step. So again, this is 3 ways of depicting it, and we're kind of in the middle. So I don't expect you to draw the structures. I do expect you to know the reactants, products, and the main enzyme. And you don't have to worry so much about cofactors or anything like that other than NADH. But you don't have to know about magnesium or ions or any of the other cofactors involved. So kind of the middle one is where we're at. So the first step we said requires the input of energy. So this is called the energy investment phase. We're actually going to burn an ATP. So phosphorylation of glucose. The first glucose molecule reacts with ATP using an enzyme called hexokinase and creates glucose 6-phosphate. Hexokinase is an enzyme that transfers a phosphate group from the ATP to the glucose 6 carbon to make glucose 6-phosphate. We said on the bottom, I'm not really going to talk so much about magnesium, but just that the magnesium is part of the reason for nucleophilic attack, for those of you that have had the organic. But all we're interested in really is that the hexokinase is an enzyme that transfers phosphate groups. The reaction mechanism is called a random by-by. You can remember when we talked about enzyme kinetics, how this works. So it means that both of them have to come onto the enzyme. So first glucose comes on, then the ATP comes on, then the reaction, the ADP goes away, and the glucose 6-phosphate is created. So the ATP binds to the hexokinase without glucose. It is not hydrolyzed by the ATP. And the reason for that is it needs both of them. The binding of glucose elicits a structural change that puts the enzyme in the correct position for the hydrolysis of the ATP. So hexokinase undergoes this conformational change that brings the ATP closer together and allows that reaction to occur. It transfers the phosphate group from the ATP to the glucose molecule. So kinases in general are enzymes that are involved in phosphorylation, and there are a number of them that we'll talk about. The hexokinase is the one we just said, and there's also a glucokinase, where hexokinase has a low Km for sugars and a broad specificity for a lot of different sugars, and it can be inhibited by glucose 6-phosphate. On the other hand, glucokinase is also involved in transferring a phosphate group to a glucose, but it has a very high Km for certain sugars and a very broad specificity for sugars as well, but it is not inhibited by glucose 6-phosphate. And in fact, this was found more predominantly in liver cells. So it elicits a different kind of response because it doesn't have the same Km. So where hexokinase gets saturated right away, and increasing activity or increasing amount of glucose in the blood, the glucokinase will pick up because hexokinase is already maxed. And you can read this. This is about a little bit more specifics about this, but I'm not going to go into it. This is just for your information about how it handles blood glucose levels. Okay. So then the next step is the conversion of glucose 6-phosphate to fructose 6-phosphate. So you can imagine that glucose and fructose are, we said, isomers of each other. So this is an isomerization reaction. It takes the glucose 6-phosphate and converts it into fructose 6-phosphate. And you can see here this is a small negative delta G. So it is not a regulatory step. It's the isomerization is critical to get the groups into the right place, but it's not a regulated step or any kind of rate determining step. And the enzyme is called phosphoglucose isomerase. The third step now is the phosphorylation of the fructose 6-phosphate to fructose 1,6-biphosphate, which means it now has 2 phosphates. So this one is also going to use a kinase and another ATP. And the kinase here is called phosphofructokinase, or PFK. And you can see here it has a large negative delta G. This is a key control step in glycolysis. It is regulated by both ADP and AMP levels, and also ATP and phosphoenolpyruvate, which is actually something further down in the chain. They actually have feedback into how this step goes along as well. And this large negative delta G is something that we want to keep track of. If we were in class, I'd be writing these on the sideboard. Okay. So it is also allosterically regulated. So the different concentrations at low ATP, that means it's needed. It's going to push this reaction forward. When there's a lot of ATP, this reaction may move a little bit slower. And when there's different combinations in between, it'll be in between. But it is a key regulatory step in glycolysis, and it induces this conformational change, and it has like this feedback. I need a pointer. It has a feedback of things further down the chain that can stop this from going on. So it is a key step. It has a large negative delta G, and it is what's called the committed step. Because at that point, you're definitely going through glycolysis. There's no turning back anymore. So step 4 is a non-hydrolytic cleavage of fructose 1,6-biphosphate. This is also not a regulatory step. And if you look at the delta G, it is a large positive delta G. So it is not favorable in the forward direction. So you can ask yourself, how does this reaction proceed? I thought things were not spontaneous if there was a positive delta G. And that can be answered. You got to think back to your general chem about Le Chatelier's principle and concentrations of substances. So when something's sort of at equilibrium, if you add a lot of one of the reactants, it'll push it towards the products. And that's what's going to happen here. When you have a lot of, I'll go back. So when you have a lot of this fructose 1,6-biphosphate coming down the pipeline because you have a lot of glucose, it's going to force it to go to the next step. We'll have another picture of this at the end. So step 5 then is the interconversion of the triose phosphates. So we have, we said that we resulted in the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And these are pretty much, they go back and forth. They interconvert very easily. The delta G's between them aren't very much, and it drives the next steps in regulation. So these are not a regulatory step either. Right? So like I said, this goes back and forth between the two different types and is this intermediate, which is like a combination of both steps. And this is proceeds by an enzyme called triose phosphate isomerase. And this would be the end of stage 1. So just looking over stage 1, we had this investment phase. We had one glucose molecule broken down into 2 glyceraldehyde 3-phosphates. And now we're going into the energy payoff phase. Each molecule will now be converted into pyruvate. Okay, I'm going to go into my other slide. Hold on a second. And I have to bring up my other slide. 1, 2. All right, maybe I'll just stop this here. And